I now call to order the Society's 2,473rd meeting in what is now the 151st year since its founding on March 13, 1871. Good evening, everyone. My name is Larry Milstein. I am the president of PSW, one of the oldest scientific societies of Washington, D.C., committed to providing a forum to further scientific understanding and inquiry. Welcome to our members, guests, and friends, including everyone here in the Powell Auditorium at the Cosmos Club, and everyone tuning in on YouTube to tonight's lecture by Andrea Ramirez. The Society is grateful to our sponsors, including PSW Full Year Series sponsors, PSW member Mike Helton and Helton Associates, and the Policy Studies Organization in cooperation with the American Public University, and the PSW Spring Lecture Series sponsor, PSW member Tim Thomas. We thank them for their generous support. Speaking of sponsors, as mentioned at previous meetings, the Society has begun a fundraising campaign to ensure that we'll be able to meet expenses in the coming years without having to reduce the number of meetings and lectures and without having to spend down its reserves. The goal was to raise $25,000 more in dues, contributions, and sponsorships than was raised in previous years. And while the campaign has had considerable success, we are still a little short of that goal. So if you can afford to contribute by sponsoring a lecture series or an individual lecture, or you just have the ability to give in any amount, please let us know. Or go to the website, homepage, and use the contribution form that you can get in one of the pull-down menus. If you have any questions, feel free to email me at president@pswscience.org. I am pleased to announce the following new members. Kevin DeCara, an undergraduate student at George Mason University, majoring in mathematics and computer science, interested in neuroscience and the emergence of complex behaviors, who learned at PSW from a friend who has attended PSW lectures. Brett Feldman, Senior Vice President at Citi, interested in physics, cosmology, game theory, and economics. Also, AI, archaeology, and Egyptology, who learned at PSW from a Sean Carroll podcast. Neil Arthur, an engineer with the National Geospatial Intelligence Agency, interested in plasma and nuclear physics and space exploration, who learned at PSW from another PSW member. And tonight's speaker, Andrea Ramirez, who learned at PSW from our very own member, Frederica Durema, and partly from the invitation to speak here tonight, and whose interest will be clear in some part from tonight's lecture. We welcome them all to membership. If you're not a member and would like to join PSW or support the Society, you can do so through the PSW website on the homepage using the blue Join button on the upper right-hand corner. We welcome new members, and we appreciate donations. Join now. All members are entitled to wear the PSW Science Rosette. The rosettes are $15 plus 90 cents DC sales tax. It can be purchased online or at the rosette table in the back, but you must pick the rosette up at a lecture here at the Cosmos Club. Recording Secretary Cameo Lance will now read the minutes of the 2,472nd meeting and the lecture by Richard Hansen, A Mayan Civilization in Northern Guatemala, revealed by extraordinary LiDAR technology. Cameo.
Thank you, Larry. And for the minutes. On February 17th, 2023, from the Powell Auditorium of the Cosmos Club in Washington, D.C., and by Zoom webinar broadcast on the PSW Science YouTube channel, President Larry Milstein called together the 2,472nd meeting of the Society at 8:01 p.m. He welcomed new members, and the recording secretary read the minutes of the previous meeting. President Milstein then introduced the two speakers for the evening, the single speaker for the evening, apologies. Richard Hansen, director of the Mirador Basin Project at the Department of Anthropology at the Idaho State University. His lecture was titled, Mayan Civilization in Northern Guatemala. Hansen began by thanking his supporters, contributors, and colleagues. He then went on to discuss the legacy of the Mirador-Calcalun-Karst Basin and delved into the history of the Mayan civilization with a focus on the pre-classic period. LiDAR technology, the speaker explained, has helped to reveal complex societies that were previously unknown. The area contains a large number of ancient cities, some of which were previously unknown. Hansen then discussed his research on the biodiversity and archaeology of a specific area within the Mirador Basin, which contains 6 types of tropical forest and is home to ancient Mayan civilization remains. The ceramics found in the area date back to 1000 BC and show intricate art and depictions of Mayan culture. The speaker went on to discuss the discovery and preservation of pre-classic art and architecture of the Mayan civilization, which showed the original colors, shapes of the buildings, elaborate water systems, and various depictions of the Mayan cosmos. The speaker explained that they leave the jungle on 3 sides of the pyramid for cost, maintenance, animal inhabitants, and future technological advancements. Hansen then reviewed the deforestation carried out by the Mayans to build thicker floors in their buildings. He pointed out that the Mayans had the same virtues and vices as us, and that their civilization collapsed due to environmental degradation caused by their activities. The speaker then explained how LiDAR technology was used to reveal the concentration and density of large early cities in the area, and how these cities are worth saving due to their size and significance. Hansen moved on to review the situation in northern Guatemala, where there is massive deforestation, logging, burning, and the intrusion of looters, poachers, and traffickers. He explains that this leads to the fragmentation of the forest, which could destroy the species that make it work. Hansen concluded by emphasizing the importance of preserving the ancient Mayan civilization in El Mirador and how to do so sustainably. He described how the area can be made accessible through a miniature train, which would allow for income to be generated through the concessions model instead of logging. The speaker explained that the ancient Mayan civilization was much larger than previously thought, with millions of inhabitants. They also speculate that the civilization may have been conquered by the Olmec civilization. Next, the question and answer period began. One of the members asked about the population and construction of the ancient Mayan civilization. The speaker responded, noting that it took 15 million man-days of labor to build the Dante Pyramid. Another member asked about getting to the sites as a tourist. The speaker commented that there is no ground transportation other than having a full guide, and one can also approach it via helicopter. After the question and answer period, President Milstein thanked the speaker, made the usual housekeeping announcements, and invited guests to join the society. President Milstein adjourned the meeting at 9:54 p.m. The temperature in Washington, D.C. was 2 degrees Celsius. The weather was partly cloudy, and in attendance, there was 78 in person. The number of online viewers in the first 2 weeks of posting was 239. Respectfully submitted, Camille Lance.
Are there any comments, suggestions, corrections on the minutes?
Motion to accept as read.
Not until I ask for it. Do I have a motion to accept as read? Thank you, Scott. Do I have a second? Thank you. Okay. All in favor? All opposed? Minutes are accepted as read and will be posted to the website in due course, subject to any necessary corrections that come to us electronically in the next couple of days. We now turn to tonight's lecture by Andrea Ramirez On the All of Us research program, NIH's plan to build a million-person health metric database. Andrea is the Chief Data Officer of the U.S. National Institute of Health's All of Us research program. In addition to her work with All of Us, Andrea maintains a clinical practice in pharmacogenomics, atypical diabetes, and general endocrinology. Before becoming the Chief Data Officer, she served as Senior Advisor to the Chief Executive Officer of the All of Us research program. And before joining NIH, she was a physician and scientist at Vanderbilt University Medical Center, studying the genomics of metabolic disorders and precision diabetes care, where she also led the data science team at the All of Us Data and Research Center. Andrea earned a BS at North Carolina State University and an MD at Duke. She completed internal medicine and clinical pharmacology training at Vanderbilt University Medical Center and endocrinology, diabetes, and metabolism at NIH. All questions will be fielded after the lecture. During the question and answer session, Andrea, the stage is yours.
Wonderful to be here with you all tonight. Thank you so much to Larry for that kind introduction. As he mentioned, I was fortunate to meet Frederica about 6 months ago at a mutual acquaintance's home in Boston and had a lovely time understanding the history of the society. So thank you all for this invitation. I hope brings some interesting things that you all can learn from, but more importantly, that I can learn from you all with your questions and feedback on the work we're doing at the NIH. I don't have conflicts of interest to report as a federal employee, financial-wise, but many, many people did contribute to this work, so thank you to them. Hopefully we'll mention some of them directly. The majority is funded by federal tax dollars, so thank you to you all. Remember, this is work of the government for the benefit of the health of the United States as well. I did go to medical school, which was mentioned, and when I was there, I had a professor, Dr. Harmel, who always talked about observation as the foundation of medicine. He really emphasized getting your hands on the patient, making sure you're looking in a good light and using your 5 senses. It was a perspective that often is lost today in medicine as we're moving much more quickly, but it really stuck with me during medical school. We also think and talked a lot about how we learned about medical ailments. And I really go back to this picture, which struck home as one of the founding principles of one of the most impactful observational cohort studies that was founded. Not for this audience, but sometimes for medical students, I ask who these guys are. I don't have to ask this audience. But in the middle is FDR. He was several months before his death at the Yalta conference when he was ailing from essential hypertension. Blood pressure over 200 was recorded routinely, and that was thought to be necessary. That was thought to be what you needed to sustain getting older, that your arteries needed to harden to continue to circulate blood. His physician, who was an otolaryngologist at the time, remarked, a combination of arterial change in blood pressure, for some reason one popped, that the arteries were strained. And he died thereafter in Georgia. And what was founded after his death in 1946 was the Framingham Heart Study. In Framingham, Massachusetts, 5,000 people were enrolled, saying, what if we just watch them? How can we observe them over time and find out, do you really need to have high blood pressure? What happens to those who have high blood pressure or have high cholesterol? Do they live longer or shorter lives? And at the time, even by 1961, some really important discoveries had been made, just a few of which I listed here. Cigarettes increase heart disease, cholesterol, blood pressure, and others. Of course, many other lines of evidence contributed to that understanding, but the Framingham Heart Study really was foundational in how we understood observing people over time and outcomes that happen. Interestingly, last year in 2022 was the 50th anniversary of the end of the Tuskegee experiment that was also an observational study understanding the outcomes of syphilis in populations that were exposed to it that actually went untreated. And this really, to me, really hits home that it took 25 years after the start of the Framingham Heart Study that we look to as a great example of learning in medicine to really end another really terrible example of engagement of participants in research where participants were not understood, they did not understand, they were not consented in participating in the research, but instead subjects of the research. So In my mind, when I think about how I learn as a physician and a scientist, two of these studies, and of course many others, really hit home about the historical perspective that studies like ours today enter into. You all know people have different disease risks, right? So everybody starts in the middle, and the job of a doctor, the job of the patient, which one am I gonna be? Am I gonna be healthy? Will I have severe disease? Will it be severe but self-limited? That really is what we're trying to do, is understand differentiating that big group in the middle. We have drugs now. How people respond to drugs is also extremely variable. You're not sure which one you're going to end up in. Are you going to get a drug that actually turns out to be more toxic than the disease itself? How can we understand, how can we observe and learn from medicine and human health to really improve those outcomes is a huge challenge science has faced for a long time. 2001, we sequenced the human genome, and many people thought, we now have the genome, we will be able to tease this apart, we will understand the DNA contribution and figure out all of this complexity. And fast forward over 20 years, we're still just at the beginning of understanding the importance of genome to science and the importance of the genome to medicine. Larry also mentioned I'm an endocrinologist, so I study diabetes and how hormones signal in the body. What I'm showing here is a study that was recently done in a large population called the UK Biobank, which has about 500,000 people enrolled. And they asked a simple question: who gets type 1 diabetes and who gets type 2 diabetes? Everyone appreciates type 1 diabetes happens in childhood, young adults, children, and type 2 diabetes is a disease of adulthood. But what this study showed when they actually observed and looked and used genetic prediction to understand who had type 1 and who had type 2 diabetes is that the red line at the bottom, here at the very bottom here, this red part is actually type 1. And you can see significant type 1 diabetes, and this is when the diabetes started, the age at diagnosis, starting in your 30s, 40s, and 50s. And that really hadn't been the understanding of type 1 and type 2 diabetes until you took the time to look at when it started and understood their underlying genetic architecture and the genetic contribution. So again, we're learning all the time how to integrate genomic data with clinical observations to get at the difference between basic diagnoses that are understood in the population. In addition to type 1 and type 2 diabetes, which is very familiar to most people, there's another form of diabetes called maturity onset diabetes of the young, where observations were made that young people, the typical form of diabetes that young people were getting was actually onsetting during maturity or at an older age. And many, many years ago, they started to recognize these huge families who had a genetically transmitted form of diabetes that could cause symptoms similar to type 1 diabetes and some similar to type 2. And over the history shown here on the right, you can see all the way from 1960 to 1975, we continued to make discoveries about monogenic diabetes in 2015 with a list of 13 genes. So this is what I do in clinic. People are sent to me in my endocrinology clinic and say, what kind of diabetes do I have? Why do I have diabetes? And even now, 60 and 70 years after a lot of these discoveries have been made, it's still an incredibly complex question. This is an example of the many different characteristics you're looking for across patients to begin to understand which category they fit into. And every category in which you're placing that patient leads to a different treatment and a different outcome. So this is just an example to show in one simple disease, diabetes, that we all understand, there's a lot of complexity, there's a lot of unknown. And it's changing all the time across different populations, the percentage and how we're observing diseases, just like a simple diagnosis like diabetes. There's a wonderful website called Diabetes Genes that started to catalog this. And there's a calculator that was developed in the UK to actually input different characteristics of participants and use databases to predict how likely it is someone has one of those atypical or different forms of diabetes. So easy, right? We know the genes, we can predict their probability that it's disease causing. But in reality, our American health system is a lot different. So as opposed to the UK or some other places where genetic testing is widely available, in our system, we're actually faced with proving a lot of cost effectiveness related to genetic testing. And a lot of studies and a lot of work by a wonderful group listed here, Dr. Naylor, who's at Chicago, have gone into proving this will improve patient outcome. It's worth it to do the testing. It can change their outcome and benefit them in a cost-effective way. This is an example of our MODI testing that we do in clinic. We can see an example bill that a participant or a patient may get in the clinic that could be almost $20,000. Often a direct-to-patient cost can be less, but you can appreciate across an entire country with a lot of inequality and a lot of different health systems to deliver, it's very difficult to deliver this level of care in many settings. So again, from this simple list, okay, we understand the genes, how can we predict who has diabetes? Every time we receive a result back from one of the genetic testing companies, an entire panel is set off that actually evaluates the implication of those mutations. So this is not to go through in detail, but to give you an appreciation that just understanding and finding a variant in DNA that may be linked in a gene we know causes a disease is not the end of the answer. There's a lot more work to be done, and many experts have input to understand how these mutations function. There's a lot more learning to be done, even though we've started to understand genomic medicine. So, extensive rule systems have also been developed and implemented through the FDA to understand how you classify a variant as pathogenic. And what we've learned as we've implemented these algorithms is that because the input of sequences, the input of people who we've actually observed to understand their underlying genetic architecture and variation, the majority are from European populations, and we've undersampled diverse communities that have different patterns of genetic variation. So that lack of diversity in genome-wide association studies shown here on the left with an almost 80% prevalence of European sequences and a much lower prevalence of non-European populations is that when we try to take individual variants and interpret them, we don't have enough data in non-European populations to correctly assign the pathogenicity. And when we try to combine many variants that may be more common and predict risk based on an outcome of what's called a polygenic risk score, we just lack the data, and your confidence intervals get really wide when you leave a European population. The data just isn't there because we haven't observed enough non-European populations to really understand the implications of the genome across a wide range of diseases. And this is just an example. That polygenic risk score that again was built in the UK Biobank population, just here trying to show from type 2 diabetes risk score to a type 1 diabetes risk score, you can see a huge overlap. We don't have enough data. We can't use genetics alone to understand and distinguish what's happening clinically in our patients. On the left, this is again showing just across one simple disease diabetes, all of the different outcomes you can see and how you really try to implement precision medicine if you're going to get there, the challenge that it will face moving into diverse populations and being able to correctly ascertain that diagnosis that's going to help your patient. So I think of outer space. I love it when they cover it in the media. I always think, man, those physicists, those astrophysicists have everything figured out. They can tell me what's happening a million light years away with such specificity, and I can't tell you what's happening to my patient with diabetes sitting right in front of me. But man, those physicists are good. So this is just an example about what makes me think of the power of observation and how we doctors need to learn more, is when the Hubble Space Telescope was introduced, one of the first pictures published in the New York Times is shown here. And people were highly critical because the left versus the right didn't seem like much of an improvement, right? That really wasn't that great. What are we doing? Why'd we spend all the money on the Hubble Space Telescope? But when you waited and let it take more pictures, the quality of the data really improved. And you had that observational time to gather the data and really begin to understand that was the foundation of understanding so much more about outer space. It hits home for me about how we're trying to understand humans, that we're really at the surface and we haven't taken the time yet to really understand the complex systems that are inputting and building that out. So fast forward 2015. President Obama saw this disparity. There was an appreciation that we didn't have the right data input, that data were growing that weren't being connected to the healthcare system, that the genome had been sequenced, but it really wasn't translating into improvements at scale in healthcare and precision medicine. And the mission was set forth by the Precision Medicine Initiative to enable a new era of medicine through research, technology, and policies that empower patients, researchers, and providers working together to develop that individualized care, figuring out why that disease is going to happen, how that medicine will respond. And this picture was always noted as one of the few times during any State of the Union that actually both sides of Congress were standing and clapping. People were very supportive when this idea was introduced. The specific goals were to recruit over 1 million diverse participants across the United States, collect health records, biosamples, blood, urine, and health surveys to find solutions for hundreds of diseases and drugs. Simple, right? Great idea, recognizing a problem, putting some money behind it to help address this problem we'd identified that really was an opportunity for America, again, to lead in discovery around medicine. So fast forward again, now we're here in 2023 with the All of Us research program. The Precision Medicine Initiative piloted several different approaches and funded what is now the All of Us research program. Again, with that central mission to accelerate health research and medical breakthroughs, enabling individualized prevention, treatment, and care for all of us. We see 3 main functions: nurturing relationships with our participants, those partnerships that will last decades with 1 million participants, reflecting the diversity of the United States, delivering one of the largest, richest biomedical data sets that's broadly available and secure, easy, right, low-hanging fruit there, and then catalyzing a whole ecosystem of communities, researchers, and funders that help make that study an indispensable part of health research. And of course, acknowledging again, as I mentioned earlier, that a huge team will be necessary to make this possible. The program developed core values at its outset to keep centered and keep true amid a lot of competing interests about what really was important. And I'll emphasize a few of them here today: that participation's open to all. I mentioned the diversity, that we really want to build trust through transparency and giving participants access to their information. To date, as of just last month, we've enrolled over 590,000 participants. That's the beginning of their journey. They complete many different steps of the program. You can see around 2020, like the rest of the world, COVID-19 impacted our enrollment. What the orange line at the top is showing is people who've come into the study, and the blue line at the bottom here is who have actually completed the initial steps of our program, generally donating their biospecimen, sharing their electronic health record, and completing surveys. We have over 430,000 biospecimens. That actually translates to almost 10 million aliquots, which are housed at the university, at Mayo Clinic, who've built one of the largest biobanks in the world to support this effort. And among those participants, we've again emphasized making sure we're inclusive of the diversity of race and ethnicities that exist in our country, as well as different UBR categories. And we define UBR as underrepresented in biomedical research, those who have not typically been included in studies. That may be because of race and ethnicity, age, sex assigned at birth, or other gender minority groups, income or geography or other categories. We're looking hard at our participants to make sure we're diverse in our recruitment. This has required a huge network that has grown across the country. Many different sites, including what we call HPO partners, or healthcare provider organizations, as well as the biobank I mentioned. We also partner with FQHC's federally qualified health centers, as well as the VA. We're also using the VA to recruit through our veterans population. A quick look across our study about how, again, we go from enrolling to gathering data. The first step is consent. So this is an human subjects research program, we do consent all participants both to participate in the study and to share their electronic health records. Right now, participants must be 18 years or older, and there's an online video consent. And again, they authorize sharing their electronic health data researchers that we're capturing right now through their healthcare provider organization, or they can link directly. Many of you may have blue button access or access through My Health or others. We can gather access to electronic health records that way as well. Participants complete surveys, basics, demographics, lifestyle, personal and family medical history. We've added social determinants of health surveys, as well as an in-depth survey about COVID-19 and participant experience, so how people were experiencing the pandemic. Recognizing a lot of other surveillance data is coming from public health databases, we're adding information about how our participants experienced the pandemic and how they interfaced to vaccination. We do gather physical measurements, blood pressure, heart rate, height and weight. These probably aren't as innovative as some studies, but can be done in a variety of settings. I mentioned the biospecimens that we've gathered so far. Most of these are banked. Primarily, we're studying the DNA, and I'll talk about our data generation in just a moment. As well as mobile and wearable device data. So we do have an ability to bring your own device. So anyone with a Fitbit, with an Apple Watch, you can link your data directly into our system. And we also gave out 10,000 Fitbit devices for people to begin to track their activity and share that data back to the program. So what's different about all of us? Why is it different? The diversity at the scale of 1 million or more people will allow us to really capture a broad range of common diseases, but also sample down into rare disease as well. We've focused on participants as partners, so moving away from a research subject model to embracing and participating with that participant throughout their journey, engaging them, and prioritizing return of value that we'll talk about shortly. And that longitudinal design. This isn't a take your data and run, this is a take your data and give us more. Participate, continue to engage. We want to gather data across the lifetime. All of these data sources are hoping to bring together not only biology, but elements of environment and lifestyle that can help tease apart the complexity underlying challenges we face in medicine. So, how do we return value to participants? That's one of the core values that we mentioned. How do participants have access to their information? Right now, as we sequence DNA, as we understand the variation in their genome, our genetics return strategy focuses partially on engagement, so things that a participant may find interesting and they may derive value from, including the things at the top, genetic ancestry and traits and results. We cover 7 regions and 21 subregions, and then some fun traits, earwax, bitter taste perception, cilantro preference. These are pretty well documented, but participants really engage well with them and think it's interesting that they can learn about themselves. The health information was much more recently released. We're looking both at hereditary disease risk as well as medicine and your DNA report. I always call it pharmacogenomics. That does not test well. No one understands what pharmacogenomics means, so we call it medicine in your DNA now. 59 genes are, by the FDA's ascertainment, returnable and actually have reportable variants. I hope you appreciated from the complexity that we look at every day in monogenic diabetes, it takes a lot of evidence to be sure something is actually causing the outcome, that you can trace it back to that specific DNA effect. So these are the diseases that we actually return genetic risk for to people. And then common drugs, CYP2C19 and others that metabolize drugs like warfarin, Plavix, other common agents, as well as a lot of antidepressants are returned as well. And participants can use that information to engage with their provider, take them back to their healthcare provider, et cetera. This is what it looks like. About over 80,000 of our participants have viewed their traits and ancestry results. And we asked some questions. Did you like it? Did you like getting this back? And many people were quite satisfied because we sequenced health data after that engagement data. Certainly their first question was, please give me the health data. But as we worked through the FDA return process, we had to get approval for that. But people have been fairly pleased with participating in that way. This is just another example of what it looks like to get that hereditary disease risk back or to get your medicine and DNA report. We just started that just this December 2022, and already I think it's up over 25,000 or so have gotten that information back. It is a pipeline, so if you started and gave your sample earlier in the study, you'll be getting your results back first. We're working through that backlog, getting up to the 100 or 200,000 or so we have in the journey now. So, researchers. It seems obvious that the data we're gathering would be valuable to researchers, but it takes a lot of design to get data at the scale we're collecting it back to actually be valuable to my previous job, which was as that researcher waiting and gathering and accessing data. And we've phrased it in our core values that the program would be a catalyst for positive change in research. What does that mean? That's very broad. We recognize a lot of challenges research has had in the past. How can we actually cause positive change? How can we accelerate that? This is a very data-centric view, but I hope you'll appreciate that on the left, a very traditional approach, many of us grew up in this world, was a central store of data existed, and many individual researchers downloaded it to their individual machine. This caused a lot of multiple copies, didn't scale to support big data, and also had security concerns because you really couldn't control the local machines people were using to house data once they downloaded it locally. What all of us has moved to is a cloud-centric approach that instead of taking data to the researchers is bringing researchers to the data. Putting all of the data centrally in a cloud that has tools available centralizes that security control, makes it accessible to all researchers. So if you're not in a well-funded lab with a huge central compute store, you can access the same compute power as everyone else in the cloud. It decreases the cost of storage and in theory lets people develop a shared tool ecosystem as well, not recreating the wheel on their individual machines as they look across data sets. This design has to be balanced against the need to protect privacy, right? Participants want the things on the bottom. They really want their data to be useful, but they also expect their data to be kept safe. All of us is committed to that broad and open access data policy while protecting participant privacy. So finding the way to do that is always aiming, as a colleague coined, sharing widely and wisely. How can we do that in a phased manner to understand we're not putting participants at risk, but we are giving researchers the data they need? Quick look at our data pipeline. As we start, the data types I described we collect, we move into a raw data repository that has extensive curation steps detailed in the middle, harmonization, privacy, and we move that data into a curated data repository. The Research Hub is then the public-facing website that researchers approach to access all of the tools we've built. A closer look at that data set, that curated data repository, has multiple different tiers across it. So the public tier that's available here is accessible on a public data browser. We'll take a look. The registered tier requires registration and a passport model we'll discuss. And in the future, we're designing ways to have individual biospecimen and participant data access that will all be built through the cloud-based Hub platform. So, public. What do we want the public to know? Who are our stakeholders engaging with these data? Definitely researchers, but also participants. People have joined the program. All of these things are consumed by not just researchers, but participants as well. So we reflect our data snapshots through consumable bytes. We have the Survey Explorer that lets you dig down and see exactly what questions were asked. And we have the data browser that shows who is in the data set and what data types might be available to match research questions. So here's a quick look at this public data browser, databrowser.researchallofus.org. As you search, you'll look across the electronic health record. All of the electronic health data is housed in a large common data model called the Observational Medicines and Outcomes Partnership. So all of the electronic health data is in individual domains and sorted in a way that can explain what's in the EHR, but is not what it looks like when your doctor puts it in. So sometimes we joke that the electronic health record when you're with your doctor is a wine glass, and when we put it into a research database, we break it into 1 million pieces, and on the backside, you have to put it back together. You have to find those pieces and piece it back together, because it's not exactly what your doctor put in the chart. You have to be able to build it back. The common data model is a common language to take many different kinds of glasses that come from lots of different electronic health records and sort it into an understandable format. When you search, say again for cancer, you may see a lot of conditions match, and we'll sort them by descending participant counts, but in the public space, these are aggregate data. You can't cross-tabulate them, so each of the rows is not individual. You may have people who have multiple diagnoses across multiple bins. You'll also be able to see the survey questions we asked, personal medical history, family health history. Those are available as well with the percentage. Just looking at these raw percentages is pretty interesting, though, and validates a lot of what we know. Skin cancer being one of the most common cancer conditions, breast cancer being a very common family health history response. We've also added genomic variant search. So I was speaking earlier with someone. You can search any gene and see all of the variants that have been found in the population. And you can also search by their predicted pathogenicity, if it's probably a variant that causes disease or not, and then sort that list by prevalence and understanding what underlying populations they come from in that public data browser. So that's the public website. That's what anyone can go see. That's how we aggregate data. But for researchers who need access to row-level data, they're guided into the Workbench. So what's the Workbench? We use a passport model. So typically, when you think about applying to get data and use it for research, you need to put in an institutional review board, a human subjects protection application, who then review exactly the question you're asking and ensure that it doesn't cause more harm than benefit to the participants in the study. But in our study, we've implemented a passport model where researchers are not approved based on their identity or their specific project application, but instead given a passport that's created by the removal of identifiers. So within our data set, we remove identifiers and allow the data set to be considered non-human subjects research. Therefore, we don't give individual study approval. We approve the researcher themselves. They're given that passport to move from the public aggregate bin data. They're given their data access approval and their passport, and they move into the Workbench where they're given the access to that participant level, row level data. Right now, the agreement and who approves that individual in our data set is their institution. So the liability that that researcher is working within our data set to the specs we've specified, that liability lies with their institution. There's an institutional agreement, and then users agree to a data user code of conduct that they will follow when interfacing with the data in our cloud. Again, it's pretty fast. I remember when the user experience people came and said, what do you think about this? It's going to take 2 hours to get access to all of us data. I said, oh my gosh, if it takes anything less than 2 months, that's a win, right? This usually takes a really long time to get access to big data sets. So to a kind of an internet user experience person, 2 hours sounded crazy. But to actually get through these steps in less than 2 hours is really, really quite a revolution for researchers. So the green check is reflecting parts the institution has to do, that institution agrees, and then 3, 4, 5, 6 are how the user proceeds through their own registration. So how's it going? So in May of 2020, just 2 months after the outbreak of the pandemic, all of us actually launched their Workbench in beta with access to the registered tier. So the registered tier was our early exploration of widely and wisely, understanding how to remove those identifiers, how to protect participant privacy. That was row-level data. Identifiers had been removed and had some additional protection. So it had a date shift, a random date shift across records, and it also had some changes to the model so that it'd be more protected against threats and less personally identifiable information in this tier. Again, that passport model was in place, and in our beta phase now, access is limited to US-based academic nonprofits and limited healthcare for-profit companies. So we found that there's extremely diverse and very longitudinal data. So what we saw when we actually pulled out the electronic health record data, even though electronic health records started, I don't know, 2005 or so, we started really entering most of our clinical data electronically after the 2007 and 2008 laws that changed. Electronic health records actually go far earlier than that because data were actually retrospectively entered into the electronic health record. So what you can see here is that across the electronic health record, for example, we have procedural and condition data that physicians have mapped all the way back to the early 80s, and we actually had to cut it off. You know, we had too much data going back and we didn't totally understand, and said, you know, for our first foray, we're going to put out what we understand so far, but really a lot more longitudinal data than expected from a very diverse base of electronic health records. In addition to that registered tier, 2 years later, in 2002, the controlled tier was launched, and that was the first release of genomic data. So the registered tier only had phenotypic data. The controlled tier in March of 2022, just about a year ago, had genotype data as well. The approach of the program was to generate array data, so individual variants read across the genome, as well as whole genome sequence data that read every base pair, not only those known variants across the genome. And that discovered 593 million unique variants, things that had never been seen before with the diversity of our population. In addition to that genotype data, we paired it again with all of the rich phenotype data. So in addition to the variant data and the genome sequence, the electronic health records were there, the Fitbit records were there, many of the surveys were there, and really started to create bands of participants with similar information. Unlike a traditional cohort study, unlike Framingham where you start with 5,000 people and all 5,000 people do the same steps of the study, our participants can choose how they participate, so we'll never be a squared off cohort. We will never have the same data across every participant. There's a lot of heterogeneity, and that's understood from the beginning in our design. In the delivery of that first genomic set about a year ago, again, the 50% diverse by ethnicity, what this chart is showing is the gray is all of the global GWAS and the percentage that was done in white populations. The blue is the All of Us study. And what this is showing is the impact that just our first set of 100,000 genomes had in increasing the availability of diverse sequencing worldwide. So a huge leap across underrepresented populations, and actually even among our white and non-white population, by those other categories that we describe people and understand their exposures over their lifetime, they were also underrepresented than traditional research participants. So what kind of research can all of us support? Everything? Is this the answer to all research? We don't know yet. We've done a lot of example studies, and I'm going to walk through some of those now. What associations between diseases can we find? Can we begin to look at why health disparities happen? Can we access those historically underrepresented populations, look at drug target discovery, and really looking to researchers for their own ideas? The cloud itself, the modalities of research, is extremely flexible. We'll show a little bit of how the tools work, but very conducive to artificial intelligence or machine learning processes, risk stratification, predictive analytics. As I mentioned, we are not a representative US sample. We've oversampled for diversity, and including and engaging populations. And we're also not a study with that uniform follow-up of all variables. Heterogeneity, missingness is built in. So what does it look like to do an association in all of us? As you enter the registered tier, as you enter the workbench, all research is housed in a workspace. When you create your workspace, you're then given access to multiple tools to help build the data set that you're looking for. Across a huge relational database, you can't possibly have a single flat file. This is a very large database. You need to understand the data structure to pull it in. That's complex. We've put some point-and-click tools in place to help get past that. As you create your workspace, importantly, you're asked to describe and give a description of the work that you'll be doing. Again, because researchers are not asked to say what they're working on, they're only approved on their identity and their institutional sign-off. Describing your work in the workspace is how the program understands the research happening. The cohort builder, concept set selector, and data set builder, again, pull all of the data out, let you start to create from the complex database what your question is, answer your hypothesis. Fueling that in the cloud are Jupyter notebooks, a very powerful programming tool that run on Python. Right now we're supporting Python 3 and R, and let you actually bring in the data that you've generated or directly use SQL and other direct query to bring it into the notebook directly. So really pretty flexible. This example of a type 2 diabetes genome-wide association study looked at 23 million SNPs. It ran in about 20 minutes, and it cost about $37 of compute. So a lot of different trade-offs in this model, but we're beginning to understand how cloud and how researchers can benefit. So what does it look like in the cohort builder? Lots of Boolean logic. You can and, you can or, you can time frame, and you'll see on the right as you go, you'll begin to aggregate depending on the attributes you add. You'll begin to see how many people you're getting. And there's your negative, so you can include and exclude and then use Boolean logic across it. When you look, there are two kind of example studies I want to walk through. Again, knowing there's some medical scientists here and some not. The genome-wide association study shown on the left will be an example of how you look at all the genotypes available. So all of someone's DNA, when you look at all of their variants, how can you say, these people have diabetes, these people don't? Let's look at all of their genotypes and see which genotypes the people with diabetes have and those who don't. That's that genome-wide approach. On the right is flipped on its head, which is a phenome-wide association study, where instead of looking at all of the genotypes available, because you have the electronic health record, you can consider that all of someone's phenotypes. So you say, I think TCF7L2 might be an important gene, that variant might be important. Let's look at the association of people with that variant and without across all of their phenotypes and see which ones might be associated with that genotype. So two studies, a genome-wide association and a phenome-wide association. The first, again, genome-wide association, we'll talk about type 2 diabetes. Phenome-wide association, we'll see how can you associate all of those different phenotypes against one locus, this one being FTO, a classic example of an obesity gene. So once you're in our interface, what we're showing here is how a researcher goes through using the cohort builder to define cases and define controls, using our dataset builder to decide what those covariates you'd like are, demographics or control demographics, pulling them all in, for example, to a type 2 diabetes or to a Jupyter notebook, using what's a Hale matrix or a way to pull in the rest of the genotype data, and then creating your covariates, merging and saving, all to run your GWAS, again, using that Hale platform in the cloud. You're then able to visualize your results. This is fed by the bucket at the Data and Research Center. Sorry, there's an acronym I didn't define. Bucket that holds all the genotype data. As you walk through, you can define your cohorts and you build them across the bottom, creating your cohorts and creating your data sets. These are then pulled in, and you can use the condition search that looks similar to the public space to find the conditions you're interested in. And then you go through, again, building out who your cases are, who your controls are, and can see if your power is going to be roughly sufficient to power the study. Again, just some demographics. Code, when you export it, even though you've used point-and-click tools, it actually gives you the queries that were generated, so you can manipulate those in the notebook as well and have access to replicate it so you don't always have to use the point-and-click tools. You have better visibility. And just a quick example, it's wonderful when notebooks look this clean. They're easy to read. Good annotation always helps. Quality control, just a classic QQ plot that says everything's looking good. And what we found was that we replicated a very known common variant signal, again, chromosome 10, highly associated with type 2 diabetes. So in summary, this was similar to the one that I'd shown before. This was on the much larger set running with over 10 million variants. And as we've evolved the study, the cost of compute on our cloud has come down. So even though this version of the GWAS was using more variants and more participants, the cost was actually less. So how about PheWAS? How does that look? How can you say what are the outcomes, what happens to different phenotypes across one genotype? A very similar flow. You're feeding in your genomic data again into the Hale matrix, but instead you're using BigQuery, generating that data set from the EHR to pull your demographics and create all the covariates, not across one phenotype, but across all of the phenotypes sorted into phe codes or different buckets for association studies. And this one just happened to be run in R. And what you're seeing here is the colors across the bottom represent all of the different phenotypes. So it goes all the way from infectious diseases across a phenotype map And those that come above the red line have a positive association, and those that are below the line do not. And what you'll appreciate, this known locus that associates with obesity also associates with traits that travel with obesity, like diabetes, sleep apnea, and hypertension, bariatric surgery. So this is often now a control tool, but also something that can be used for discovery in variants that may not have been seen before or at a rate high enough to appreciate at the population level. Lots of diseases. There was always a power calculation saying, why a million? What's a million, right? It's just a number that sounds good, but these are estimates of the case prevalences that may be there at scale. And in total, the goal, again, to really change and catalyze positive change in research is to lower barriers and accelerate research, not just by ourselves in the cohort or in the consortium, but enabling that research for others. So we are attempting to take those petabytes of data and bring down the cost, create common tools, and also build in research support. Within all of the tools I showed you, there's a help desk anyone can click through and ask questions to people who are sitting at Vanderbilt waiting to answer questions. These are real people. We haven't gotten to AI answered questions yet. It's actually just people trying to learn about the data set. So who's using this? We have over 4,000 registered researchers, over 5,000 projects, and hundreds of publications. There's been a focus on engaging institutions that also represent underrepresented researchers, HBCUs and HSIs. And we've seen, in addition to academic institutions, as I mentioned, because we're limited to not-for-profits right now, we have 64 healthcare organizations, that's our only for-profit group, and not-for-profit organizations, as well as federal or other government entities that have a slightly different legal agreement. They're sorted differently. So who are our researchers? We're looking carefully to understand who our researchers are as well, not only by their race and ethnicity, by their career level. You can appreciate how we want to engage and bring people in early in their career as well. So, as I mentioned, when you open your notebook, when you open that cohort workspace, you're asked to describe your research. The one thing I didn't mention that's on our public hub is the researcher projects directory. So this research project directory, every single workspace that a researcher creates actually has the description posted publicly. So anyone can go in and see what work is going on in all of us and understand the projects that are there. This full list of research projects is really fascinating. You can search by name, you can search by subject to understand who's working on what. This is a shift from research where researchers hid in their corner and didn't tell anyone what they were working on. We're trying to bring this out, get people to collaborate, and again, as a tool to engage our participants so they can also see how their contribution is changing science. Lots of papers, lots more papers, really interesting looks, some looking at racial and ethnic differences, some looking at cancer, just a lot of really interesting work. We'll talk a little bit more about some. We're tracking metrics, and we're seeing a lot of engagement across articles that are coming out of all of us as well. There's more data coming, so it's March of 2023. In the registered tier, we're continuing to provide that lower level of access that doesn't include genomic data. Many people don't want to access genomic data, so the registered tier remains. Controlled tier, we're expanding demographics, we're giving additional COVID-19 data, and really rolling out a huge growth in the genomic data. And what does that look like? So next month, about a month from now, the next tranche of genome data is coming. We're going from what have been 98,000 whole genome sequences to almost 250,000 whole genome sequences available. We're also adding a new technology that I know many of you just from dinner conversation are familiar with, long-read technology. The majority of our whole genome sequence is generated using relatively short reads that we align across the genome and use statistical probability to predict what that read means. Long read is a different technology, actually stretches much farther and has a different ability to look at some highly repetitive regions of the genome. Those 1,000 samples are the beginning of a planned 10,000 pilot. And we're also beginning to call structural variants. So it's familiar to think of a single genotype change, but structural variants may look at a larger region of the genome not captured by a single variant that could also associate to disease. This will be the largest whole genome sequence data set in the world available for broad use at this time when it's released next month. We're really excited and excited to get more researchers involved in using this data set. So, future, where are we going? How do we actually take all the participants who are engaged and let researchers not just use their data, but recontact those participants, ask more questions? So when I first came to the program, I worked on strategy for ancillary studies, and what does that look like? How do we go from what's been a startup, proof of principle, proof of concept, can we do this? Can we build this ecosystem? How should we recontact participants? How should we use those biospecimens? How we've defined an ancillary study as a partnership that expands the data set, either adding participants we hadn't targeted or adding new data that was outside the planned roadmap. We're not planning for this to be a research registry where participants leave our ecosystem because we want all of that data to come back. It's important for us to continue to capture value for researchers in our set. And we know that examples researchers are looking for are custom questions. I made a discovery on 10 people. Can I call them? Why? What's different about those 10 people? You didn't ask the right question. Let me ask more. We're looking to build that functionality in. We've piloted several partnerships, one looking at pregnant patients called PregSource, one that I'll talk a little bit more in detail where we looked at COVID signals available in our serology data. And we see growth. You know, now here we are. What this big green line is where we hope to get by the end of 2026 in reaching our million. While we pilot our partnerships and grow over time, hoping to launch studies, we want to be ready to engage with researchers at scale when we've recruited the million or more. When we get there and have understood engagement and bringing participants in, can we be ready to help researchers answer those deeper questions? That's our goal. The COVID serology study I referred to was quite interesting. We were recruiting from January through March of 2020. We stopped recruitment when the rest of the country shut down. And that meant that we had almost 30,000 samples sitting in the biobank. So we went back and did antibody studies, not one. I think the general public understands that looking for an antibody doesn't mean disease. We looked at actually 5 different types of antibodies to predict who may have been exposed to COVID-19 in their state before their first publicly related case. And what we found were 9 cases in 5 states, 7 that were earlier than known cases in those states. You can appreciate calling with confidence anything of such a small count within a much larger data set of 25,000 samples is very difficult. But we were also able to return those results to participants, building on that engagement model that when we have data that participants may find valuable, we'll give it back to them as well. So this was a really interesting study, starting to show how all of us and other studies contributed. It's important that research also be findable, accessible, interoperable, and reusable, an acronym being known as FAIR. That FAIR acronym, who may be familiar with Jupyter Notebooks, the beauty of Jupyter Notebooks in the cloud is that you can actually leave your artifact for replication anytime. So we're looking to make Jupyter Notebooks available at scale in the cloud. So when you see that wonderful paper the postdoc at Harvard published, you can go into our Workbench, pull down that notebook, copy it, replicate it, and challenge it. Understand, build on it, that you're not going to start from scratch and that you're not going to question their methodology because it's all there, it's all marked down. We've been building example demonstration projects to this effect. We did this with the COVID serology study. Any researcher in the Workbench can go in and see the exact code used to generate the figures in the published paper. Nutrition for Precision Health is a really exciting effort launching now. This is one of our first large-scale ancillary studies that's going to take 10,000 All of Us participants and do in-depth dietary studies. That does involve liquid meal replacement, so beware. I don't recommend enrolling. No, they'll be mad if I say that, but there will be meal studies. But we're looking to understand and build predictive models of who responds and how genome interacts with dietary supplements. Happening right now, this should launch around the end of this month. We're adding this study on to All of Us in a powered by All of Us model to understand again, how can we engage, how can we move through that study. In addition to the In addition to the science that we're doing, we recognize that we've gone through the first 5 years of all of us. We need to know what's going to happen in the next 5 years. And to that end, we built a 5-year goal structure aimed at the areas you see here, enrollment and retention. We've got to get there. We need to get this to scale. Getting the data available to researchers, launching ancillary studies I discussed, and supporting that diverse global community of researchers. We're open to US-based institutions now. We want to grow that globally. Incorporating that participant return of value, we talked about return of results. How can participants stay engaged? How can we return value as well? We know there's a lot more future scientific capabilities. We're again using this demonstration project model to not replace the work of scientists, but understand and build on how we can support their work. So what are demonstration projects? Why are we doing them? We've been challenged and funded not to make the discoveries, but support the community's use of data. So when we go in to do a demonstration project, we're looking to characterize and validate the cohort for quality, utility, and diversity of data and tools, not make those first discoveries. But doing those demonstration projects really help researchers understand how to use it, what's there, and help us self-identify our own quality issues or changes in the data set we may be seeing. Back in November 2019, we thought this was a lot of people. Right before the pandemic, we met and launched a series of demonstration projects to get at how the data structure works and how we can support research use. So, one of our first papers that came out was looking again to describe the data set. And how we're telling people about our data set is to describe that diversity, really looking at those underrepresented groups and showing who qualifies or who has been identified in an underrepresented group and comparing them against those who have not. So all of this data, all of the code used to generate and identify these cohorts, researchers have been able to access and not have to redo it themselves, not start from scratch. Another example, we've always appreciated gender disparities, racial and ethnic and gender disparities in obesity. We were recognizing all of us as not a representative cohort, comparing against some well-known large cohorts to see how similar or dissimilar we are using NHANES data was very powerful to see and understand the data complexity and the data quality we were seeing from the many, many different sources across the country. We did not recruit kids, right? We didn't. We only recruited people who were over 18 years old. But we went back in the data set and said, but we have all of this electronic health record data. How can we identify pediatric data to use for pediatric research? Even in our first release done several years ago now, we had almost 20,000 participants with pediatric data. I really like the Venn diagram this student generated, showing here that, remember, the electronic health record isn't one thing. Every electronic health record will have different data types. And what you're seeing is, again, that common data model broken down by different data types, and understanding that just because you have the EHR, your data type difference is going to be really great across each record. And again, just an unexpected, really, depth of data going back, seeing many different sites, if you break it down by many, many different sites sending us data, how heterogeneous it is across the data and the depth of data that are provided. So an example case we used in pediatrics was looking at pediatric obesity. We hypothesized it had been changing over time, and we're able to confirm that trend in diagnoses in our data and look at it across other common diagnoses to understand, using a statistical correction, that this was more than you would expect, that this was changing more than the others as well. So now we're still doing these demonstration projects. Those were some early ones we did just on the phenotype data. We've moved into the genotype data. I described for you the difference between doing a PheWAS and a GWAS, or a GWAS where you're looking at a gene across phenotypes and a phenotype with one gene, or gene across all of the phenotypes. You can do that on both sides. So you can do a huge study looking at every phenotype across every genotype. We're generating that data because it will cost at scale hundreds of thousands of dollars, making those results available to researchers soon in what's called kind of a PheWAS by GWAS, or we call it the all by all. We're looking at building polygenic risk scores to actually diversify the database that's been used. We hadn't had good representation outside European populations, making those covariates, making that data available. And then sequencing pilot data, that long read that we're doing, is really important to understand how much should we invest in new technology do we need to move and change that platform? So we're looking at integrating new data types as well, our American Indian Alaska Native population and the unique engagement opportunities there, and then understanding the social determinants of health data that we're gathering as well. These demonstration projects are trying to really pull down computational burden and make the results available at scale, giving everyone the accessibility and the transparency to use all of the notebooks generating the output. So what do I do now? As I came from academia, we established our Office of Data and Analytics. We sit very close to the Office of the CEO, and our goal, as we've described it, is to really maximize not just the scientific value that I've focused on over the last hour, but also the analytic value. We've moved from a startup proof of concept, and we have a challenging 5, hopefully 10, 50 years ahead. How can we use our data to predict how to be more efficient, how to engage our participants, and really maximize that analytic value that will lead to sustainable model and contribution, both returning value to participants, but fulfilling our value for health discovery as well. So some of the people I work with a lot of people. This is several of them in my own team that I've gotten to know very well and really appreciate the expertise they bring every day in service, again, of this very large public program. So the next thing we're doing, I think the most exciting thing we're doing right now, and I apologize, I didn't realize how the years don't show up here, but we're here now. This is V7. The data set I discussed today being released as V7. We have plans going all the way out to get to scale at V11, right? When, we'll all win the lottery and retire, right? But this is how we're going to get there. How can we actually go? Are the tools that we have going to scale? Can we actually use these data types, and how will we be able to integrate new data types? Laying out this plan, laying out that strategy has been a real challenge and opportunity for the program when we're moving out of, again, that startup proof of principle into maximizing the value we can return. We have a lot of research funding opportunities. I'm never sure who's looking for funding, but I like to let people know. We are giving out small grants. We are giving out enhanced ability to use the data as well. We have a few new funding notices out as well, if anyone is interested in very large-scale research. I can't emphasize enough how important it has been to engage communities and trusted partners. I didn't emphasize it as much tonight, but our huge engagement ecosystem has actually made this work, has actually been there on the ground talking to communities, making sure they understand how we're using data. Building that trust, building that transparency has been a huge benefit and proof of success in the program. Many, many people contribute. Again, our frontline staff, those community partners, some of whom you saw, all of these come together to build an engaged model that's much different than the history and other systems that were built into traditional research. There's an event coming up very soon, May 8th to 12th. We actually are going to have the All of Us Journey bus at the NIH campus. So if anyone wants a day trip to Bethesda, the study will be enrolling on that day, that time in May, right in the middle, in front of the clinical center as well. This is a funny quote, again, back to my days staring at stars, that I like to think, we've sought firm ground and found none. The deeper we penetrate, more restless becomes the universe. All is rushing about and vibrating in a wild dance. Again, built not for medical science, but it's how I think about medical science. So hopefully all of us will form that foundation to really improve and understand the complexity at scale for physiology as well. So this is not my work. This is Dr. Flores at Boston. He's at Harvard, wonderful scientist. They have a vision where we can go from those clinical-based categories, integrating data at scale and building data-driven clusters. This is the vision. This is what every diabetologist, every endocrinologist wants to get to improve human health. I hope all of us, and I hope the work we're doing and all of us will really support this vision across a range of diseases and get us to understand how we can improve human health and make use of all the data being generated as well. So thank you to our partners, all of our participants, and all of you for your attention. So thank you.
Okay, we have time for questions, and we have this procedure for questions, which I see some of you know.
Bob Hershey, I'm a member. What diseases are likely to be better treated as a result of these studies?
We are attempting to be disease agnostic. So we're building this to provide data across all diseases, at least those being captured by the electronic health record. Right now, I'd say diseases that are well captured by the electronic health record will be better represented than those outside the medical system right now. But in the future, we're building other observational methods as well. So hopefully, all of them.
Red microphone?
I'm Raj. I'm not a member. I have a lot of questions, so I'm going to ask a couple of them.
You can have 2,
Two? Okay.
maybe 3, if they're good questions.
Yeah. How do you go about maintaining privacy safeguards? Should anybody be concerned about potentially this information being shared with any insurance companies? My other question is...
Wait, let's take them one at a time.
Okay.
Sure. So it's a great question. We think about it a lot. We talk to participants about it explicitly in our consent. We put every safeguard we can, but as any research study, harm may always come to participants. So I never want any research participant to think any research study is 100% safe, but we're doing our best. And many security controls that have been put in place, again, focus on that cloud-based centralization of data. So we know exactly who's coming into the cloud and when data are going out, because we've put in place efflux monitoring. Right now, we're not approving anyone to come in and use it for those purposes. So if they were using it for those purposes, that would be in violation of our data user code of conduct, and we can actually have several large sticks that are punishment arms, as well as restricting their data. So through the cloud-based model, restricting who comes in and watching who goes out, as well as monitoring what they're using data for, we're trying to guard against that, in addition to the public safety mechanisms put in place through the Genetic Information Act and several other policy mechanisms. So, great question.
Yeah, I forgot to say that this is just fascinating.
Yeah.
Thank you for the presentation.
Sure.
My other question is, I'm looking at it as a patient. I don't know much about genomic data. I don't even know what actually it constitutes as a layperson. So I'm looking at it as a patient. So if I were to enroll, if I'm interested in, let's say I'm interested in the medical component of it as well. Okay, based on my genes, okay, if I'm prone to X, Y, and Z, would there be this medical component where somebody from this team, a medical practitioner, would help me as well?
Great question. So the line between research and clinical care is fine. We want to distinguish what we're providing back as return of value and what replaces clinical care. As a clinician, I'm always sensitive to creating what's considered a patient-clinician relationship or a participant-clinician relationship. In our study model right now, we're not looking at returning diagnostic information beyond what is FDA approved and being sequenced in the genome. So we're looking at the DNA and looking at variants that the FDA has agreed that variant means one thing only and it needs clinical follow-up. We're not doing the same thing with electronic health records, where if we see something in the electronic health record, we have not built approved FDA oversight mechanisms to say, we think somebody missed something in your electronic health record. We're returning your electronic health record so you can see it yourself in an interface, but we're not making inferences or judgments based on your clinical information in the same fashion we are in the genome. Does that make sense? Enough? I can go back a stage too. Yeah, okay, yeah, I'm happy to. It's a great question.
Let me ask a follow-up question to the first question, and that is, to what extent are you stripping out identifiers and assigning things like a random number to the patient identifiers so that they can't be traced back to the patient unless certain other procedures are invoked? Are you doing that? Or?
Yes, so each data set we release has a patient identifier that's unique, a participant identifier. That participant ID is different from one data set to the next, so you actually don't track between data sets. And the information itself has a random generated hashed identifier that actually doesn't go back through the data set. So we generate it on each data set, not backwards compatible across them. And I can go a little bit more into detail on that. So the nuance there is, you're participating in our study, you're consenting to participate. You can choose not to participate. If you choose not to participate, if you withdraw from the study, you will also be removed from the data set. So the only way we can guarantee removal of that participant from a future data set is by leaving them in the past data set. And that's so that past data set is still fair, findable, accessible, interoperable, and reusable, that we don't take people out while they're participating, but in the future they're removed in future generated data sets. And that's how the hashed identifier is not the same. We're regenerating that for each set.
I guess the other question was, do you have any plans to allow participants to select the information that you feed back to them? So that,
Actually, yeah, I'm sorry, I cut you off.
no, go ahead.
Every time you get information back, at the beginning of your return process, there's an informing loop. So the beginning of the study, you consent to participate, and participating includes return of results. But every time you're offered a result, there's an informing loop on the front that says, we're about to tell you your genome. Your genome information is available. Do you want it? This is what it is. This is how you would understand it. And you're given the option to be informed before you get that result on everything from serology all the way through to genotype information.
Brad Galvin, member. A few questions. First one, obviously there's going to be a lot of interest to use machine learning, AI, or different variations with this data. But of course, we all know that the quality of that data matters for the output that you get. So with, you're pulling in quite a bit of the electronic, medical records. Physicians don't always use the same ontology, right, to report whatever the symptoms or the disease might be. Plus you've got surveys, right, from the individuals themselves. So how are you handling the medical ontology aspect to make sure things are binned correctly?
It's a great question. Thank you for that question. So when you think about specificity, right, and you want to generate a 2x2 and see what your agreement is, you often think about just having a yes and a no and a yes and a no to see what matches. But because of our underlying data structure and the missingness, you end up with many different choices. Either never had it, have it but it's not valid, have it but it's not complete, have a little bit, have more. So you actually generate many different, what we actually built out in theory as a 6x6 about agreement in different data sources. And we built some of those examples out because just saying who has diabetes, how are you going to assign that diagnosis, differs for every hypothesis test. So if your hypothesis you're testing, you want a huge case study, a huge population, it's going to be robust statistically to missingness depending on the question you're asking, you can choose in that overlap to say, or, I'm going to say anybody or in their electronic health record or survey or even their genome, if they're thought to have diabetes, I'll put them in. But other hypotheses that are really depend on that specificity and getting it right, we're building out matrices to actually say, hey, we're really confident, we know this person really had diabetes. It really is hypothesis dependent, and I know that's not a great answer, but by providing the data and letting people build those methods is going to build out how we use those data types. And I think we're still at the beginning of how robust some of the machine learning and certainly some of the AI will be to such differentially captured data. Making sure the metadata is available, I think, is very important. We've done that with the electronic health records. We're doing that in the surveys as well. We capture surveys in different ways. Understanding that metadata implication, I think, will be very important to avoiding false conclusions, both type 1 and type 2 error across the data set.
Yeah, because some of that feedback is going to be very subjective, right?
Correct.
So someone could have the same symptoms, but they could scale it differently, which means it introduces noise, right, into potentially the conclusions that may come out of some of the AI-based methodologies. Mm-hmm. So, you mentioned you were doing manual curation, or you are curating, I should say. Mm-hmm. Are you using natural language processing to help with that, or is this all manual curation that's being done?
We're starting to. So most of the electronic health record data right now that we're providing to researchers is structured. So it's billing codes, it's not free text. We're exploring pilots of natural language processing, extracting concepts from notes. Not there yet. Another thing that you should all think about is how medical records and what your doctor puts in your record has changed over time. Mm-hmm. Appreciating the change over time when you go from one record to another, when you go in and out of different legal environments, when you go into now where all patients read your medical records, right? Just as a clinician, I think my notes are extremely different that I write today when I know my patient's going to read that note the next day than I wrote 20 years ago when it had lots of medical data in it and lots of medical terms. I write things that are much more patient-friendly now because they can see the note tomorrow. So this will be the beginning of research on data like that to understand where you can make mistakes, and that's why replication and validation, I think, are really important early on.
So a long way to go.
I think so. Hopefully, we want a long way to go.
Could you speak up a bit, Brad?
Sorry, next question. You picked 7 of the drug metabolizing enzymes, the 2C19 being one of them, with 3A4 being one of the major metabolizing enzymes. Why wasn't that on the list?
I missed that last word.
Why 3A4 was not on the list of your metabolizing?
So we're going back and forth to the FDA all the time to add those. And so we're using an FDA oversight process to add it. We're certainly looking at it, and we have called all of the pharmacogenomic variants that require different variant calling software than a standard call because of the allele structure. You have to call the alleles across pharmacovariants differently. We've made that data set available in the researcher workbench. On the patient participant return side, that goes through the FDA first, but we're working all the time with them on that process.
Okay, and then last question, the serum samples you're collecting, right, from these individuals, are they going to be broadly available? You said to potentially academics, but are there also opportunities for for-profit companies to be involved? Still make the data, publicly available, but participate, number one. Number two, when you're storing those samples, are they aliquoted out, or are you having to go through multiple freeze-thaw?
They're aliquoted.
Okay.
And we absolutely have in-scope broad availability. We've started our widely and wisely with academics, but absolutely in-scope in the vision for ancillary studies is engaging for-profit and others. Interestingly, when you engage with communities and you ask participants in some settings, do you want drug companies to have access to your data? Depending how you ask the question, many participants answer, of course I do, otherwise who's going to make the drug? So we don't see this as a negative. We see it as understanding and engaging with participants to see the value and returning data to better health. It's not a zero-sum game, and there's a lot of opportunity there the program's been committed to.
I'm Timothy Thomas. I'm a member of the Society. My question is about the text database that you mentioned a minute ago. Long time ago, I had an opportunity to analyze a large, for that time, tens of thousands of patient encounters, pure text, no structured data at all. And we found it extraordinarily rich. I'm wondering whether, extremely difficult to get useful information out of, extremely difficult, particularly to get rid of personally identifying information. Because it's pretty easy to guess who a physician's talking about. But I'm wondering if you're keeping all that data, because it's actually, I believe, the richest source of community health data available, not the structured data. Because the structured data is mostly, how do we bill?
Yep.
Do I call this a headache or a tumor or whatever, right?
Yeah.
So I wonder if you're giving enough energy to the text.
Yep. So absolutely, it's a great point. So an early study we did at Vanderbilt in the electronic health record was looking for an association with rheumatoid arthritis and psoriatic arthritis that had a known genetic signal. And we couldn't find it because you don't code for psoriatic arthritis, you code for rheumatoid arthritis because that's reimbursed, and that's how patients get their drug covered. So certainly the electronic health record is a billing tool. It is certainly gone that way much more so over the last 10 years, and that content of text notes has also changed. So if you just quantify the number of words a doctor wrote, if I look at notes I wrote in 2005 and notes I'm writing in 2022, I used to dictate, right? I would dictate and talk all day and get it back and sign my note. No more. Notes are very short, they're very abbreviated, and they're also talking to the patient now because our notes are open. My note used to talk about the patient in the third person. Sometimes now in my notes, I know Sally's reading it tomorrow. I'm just talking to her. I'm like, remember what we said, take your pill in the morning, right? My notes are communicating to the patients now. So I think it's a very rich source. Another just clinical care challenge is, does that note still communicate to other physicians? And there's a whole nother side of medical documentation where we're not using the note, we're using a communication. So if I need to tell Sally's other doctor, transplant doctor, something, I send a separate communication that may not be in the note because I have to talk to the other doctor. The note used to be what doctors talk to each other through. The note's becoming what we talk to the patient through, and that's a huge opportunity to understand how transmission of information is changing. I completely agree. We're capturing it, we're consenting for it, but there's a huge opportunity to learn more. Yes, yes, well, we have the ability to get it all. We have the ability to get it all. We haven't necessarily pulled it in yet because we store data federated and then we pull data types on an ETL cycle. So we have the access to it. We haven't organized it yet to give to researchers.
Gary List, Johns Hopkins University APL, a member. If I'm a participant in this
Speak
study,
up.
I'm a participant in this study, and as an educated layman, but certainly not an expert, I'd like to know where my values, where my genotype, my phenotype lies compared to the public at large. Am I getting old normally? Am I falling apart in different ways? Is there a way to help that a non-expert can learn what their health situation is compared to the population at large?
We're looking at tools that will put the background signal in the app. So I didn't show the app pictures today, but everything you do to enroll is through an All of Us app on a phone or that runs on the internet. Most of the return are individual results, not against a population background. All of our data go through the IRB. Well, researchers don't have IRB approval. Certainly everything we show participants goes through the IRB. So we're testing different mechanisms and different technologies to give that background signal. Some of the results have it, like for example, the COVID study described the overall findings in addition to the individual, but some don't. For example, when we capture physical measurements of height and weight, and we give someone a BMI, we don't actually show a population BMI yet because we have to prove to the IRB that we're explaining it well enough that participants can understand it, and we're not quite there yet, but it's a great question. We're trying to think about that context as well.
It would be very nice as a possible consumer,
Speak up,
a member in your study, I would like that.
Yeah.
So from the web, Jorge Apont of Colombia, South America asks, how can private companies add value to this database?
How can they add value? When they have access, we're looking for them to use it. So again, All of Us has stated publicly, the goal has always been to expand researcher access, and everything we do aims for all researchers using the data to take their notebook, not only publish in peer-reviewed literature, but make that notebook findable, accessible, interoperable, and reusable so other researchers can learn from it. So I encourage all researchers to aim for that standard, and the Workbench allows those notebooks to be shared. I think that's the nearest term thing when access is there, and then support when we do have ancillary studies available to come in and help support with the hypotheses needed, help us understand from private industry what they're looking for as well will be a two-way street.
Okay.
I'm Federica Dorima, member. Thank you for your very enlightening presentation, very interesting efforts and valuable, I think. So you talked about wanting to expand the data, and you talked also about using other data, like, the DoD data for veterans and so on. And well, Larry wants us to ask only one question, so I'm going to ask a question that has several items. So the other, and then you said your databases right now have very smaller, relatively smaller numbers for Asian, other populations. So sources for SATs are in other countries. Do you see the role of WHO, the World Health Organization, that maybe have access to such databases? And of course, then the technical problem rises, no common data models, but also there, the AI methods can, in a sense, help to kind of create common data models across the data. So what are your thoughts about this?
Yeah, it's the functionality I didn't emphasize, but that the Workbench does support is uploading any data set you're able to access from other places. You can import that into our Workbench. You can also query against outside data sources within the Workbench. And we are building some pilots because as we're consented, we're building, they're called trees, trusted research environments. And our own Workbench is a trusted research environment, how do we make it work with the UK Biobank's trusted research environment? How do we make it work with the Million Veteran Program trusted research environment that stays true to the consent and policies in place for all of those? Is that going to be through a meta-analysis where you pull down, pull down common data and analyze outside those environments? Or is that going to be through a federated program where you're actually able to directly access, or a neutral Switzerland model where we get everybody to agree we're going to share? Those are big opportunities that we haven't addressed yet. We're getting them right, I think, in our own fiefdoms, but the real North Star, either that Switzerland model or moving to a place where you can federate in a much more intelligent way that's being piloted right now at our Genome Center. So hopefully we'll get there. It's a huge opportunity.
So I did want to ask you, Framingham was a really important study. We're still learning things from it. So how would you contrast this study with the Framingham study? What are the differences in their methodology compared to yours?
Great question. So when I was, I don't know, what was I? I was a medical student. The first thing I genotyped was samples from the Framingham heart study. I sent a letter, I sent an application, they sent me plasma, and I genotyped it. So it's a wonderful study. It is very, by today's standards, small scale, yet has still generated really important data. So we think about this a lot at All of Us, which is what is the reason for big data? And are most people coming in and using our data, using it for its volume or using it for its specificity? We're still in a time where many of the studies are case control, where many people are coming in and doing case control studies in a bivariate way, very similar to Framingham. The challenge is we aren't built like Framingham. We are not complete. We are not squared off. We have a lot of missingness. So making sure researchers aren't missing confounders, they aren't drawing associations and conclusions improperly, I think is where methodology developed for traditional prospective longitudinal cohorts like Framingham will not apply, and we need to be careful about our model, which is cross-sectional, prospective, and retrospective. So when you enroll in our study, we're gathering retrospective data, cross-sectional data, and prospective data. So the methodology is different than Framingham that was started and followed prospectively and has a much more complete model. Their follow-up rate is over 98%, I think last time I looked. Ours, 50%. We're getting 50% response. We're incentivizing it differently. We're engaging people differently and really looking at that. So there's a lot of similarities and a lot of differences that we're building out what the appropriate research questions will be for our model as well.
Did they have a lot more control over their diagnostic profiles and their diagnostic toolkit, or was it similar to this where there are a lot of individual physicians who were gathering data and sending it back? I was under the impression they were very controlled.
Correct. It was very controlled. They did direct assays. So when you had a hemoglobin A1C pulled down in a data set from Framingham, it was measured the same way. They used the same assay, the same time, the same lab across all 5,000 samples or the larger cohorts that came on in the 2 offspring cohorts. All of us right now, we're using electronic health record data. So an A1C is a hemoglobin A1C that measures average blood glucose over 3 months, is a very common clinical test. That's not just one clinical test. It's mapped into a common data model fueled by an informatics system called LOINC codes. So if you're looking for an A1C, there's hundreds of different A1C tests that have been done. So how you clean that data, how you look, and again, the hypothesis you're testing, if you're trying to say which drug works best or can I approve a drug on FDA standards, we're certainly not going to have the specificity with real world evidence to do that. But if you want to know if someone has diabetes, an A1C of 10, no matter what test it's on, is probably diabetes. An A1C of 6.5 versus 6 to understand drug response, probably not in our real world data near term as good of an answer.
So I'm going to ask 2 more, and then we'll go to the audience. So be patient with me. How deep does your metadata go for kits like, for tests like the A1C test? Does the database contain, like we did A1C test using the CellX test that was lot number XYZ carried out by such and so lab. Does it go that deep, or is it just they did an A1C test?
It varies by site. So it varies by electronic health site, and as we talked about, it also varies by time. So many electronic health records capture an A1C from 1998 because they have the number, 6.5, but won't have any of that metadata. But when they moved into newer versions of electronic health records and information knowledge management systems, they have a lot more data. So again, different across every site, different across multiple labs and multiple sources. We capture as much as we can. It's provided in the data model, but every researcher has to know their hypothesis, describe, clean, and look at the data to make sure they're not drawing false conclusions.
Are you aiming to have that deep data?
We're
Or
considering
just taking it if you get it?
Yeah, no, we're considering it. So the question of generating assays on our samples, we're not doing it right now as they come into the biobank. So you would be pulling a sample from storage. And there's a team looking very carefully at that right now, led by Angie Music at the program, to think, what is the right question to ask? What's going to both return value to participants, add value of what's not available in the electronic health record, and also benefit a research question. So, for example, total cholesterol is measured really frequently on the electronic health record. So probably you're not going to gain as much by measuring total cholesterol. But lipoprotein A, a really important marker for cardiovascular disease, is not often measured clinically. Maybe we should measure lipoprotein A. It has a lot of benefit to population to return to stratify risk that's not well understood in some populations. But if you measure lipoprotein A, you can probably measure 20 other things for the same price. So they're looking very carefully at balancing what to measure, what's already there, and what's going to return both research value and possibly clinical value to the patient.
So my last question is a little more difficult, I think. So we use very crude bins for dividing people up demographically. We got white and we got Asian. I mean, those are very crude. And I wonder if you see in the genomic data that you're getting, and maybe less importantly in the phenotypic data, whether there are any clusterings that allow a more precise demographic binning of populations that would be more meaningful to medical care?
Sure. So ancestry, there's a lot more ancestry in genomic data, and we can understand ancestry in many different ways. So ethnicity and race are different than ancestry. Our surveys, what I've reported, are what we call the high-level categories of race and ethnicity. There was a breakout question you can see in our Survey Explorer where people also answered individual categories of how they self-identify within those larger buckets. That's available as well. Within ancestry, ancestry is used in research for different things, to either create principal components to adjust and pull out confounders, because if you don't pull out ancestry from analyses, you're going to skew your results. So you want to control for ancestry at a high level. There are many different methodologies being used to ascertain that ancestry and to figure out if you're using the right ancestry or not. One of our best demonstration projects, led by King Jordan and others out of Georgia Tech and other Medical College of Georgia as well, were building out different algorithms to show how we should be assigning ancestry. You can blind your genomic analysis to self-reported ancestry, or self-reported race and ethnicity, or you can look at it and bias it by that. There's a lot of methodologic discovery to be done to understand which one is better to pull out confounders and to clean your analysis. So a lot more data there, methodologic development. Being the largest data set at scale, 250,000 genomes coming this month, it's one of the first times it's been done at this scale in a diverse population, so a lot more to learn.
We'll have the red microphone.
Hello, yes, good evening everyone, excellent presentation. I actually work...
Name and whether you're a member.
No, I'm a guest.
And name? Name?
Noel Watson.
Thank you.
Okay. I work at NIH in CIT, so I'm delighted to be here. We get lots, I'm going to keep my questions really short. The calls that we get a lot are people who are registering. I remember one guy recently was from a university, but he's not a US citizen, or he's visiting. And part of the registration process required a state-issued ID. And we didn't know what to do except to direct him to whatever support information was there for the All of Us program. If you maybe can speak to that a little bit.
Yes, so identity verification is an opportunity and a challenge for the program, both in identifying participants, so understanding who our participants are, and in identifying researchers. So I'm not sure, I think you might be talking about researchers. Yes, so researchers, we're looking at a lot of unique ways. So that non-US resident right now, if they're based at a US-based institution, again, their approval's based on the US-based institution. Their individual identity verification then has to proceed differently, and there are some mechanisms in the help desk for that. But how that transitions into international access is being addressed right now. Again, a goal for the program, working through how to make that work, and identity verification is a huge issue because, again, right now, the institutions are accepting the liability, but the consequences also come on the individual basis. So understanding who that individual is of the highest importance. Some methods work really well for most people, but we have those individual processes to use, and we're trying to learn in the NIH ecosystem. I'm sure you know this is too detailed, but that we're not using the same method lots of other studies use, and we're trying to make sure we're both contributing back, but not being blind if something else is working better as we're looking at international. But yeah, great to be here. Thanks.
We're going to go to the blue microphone, and then we'll go to the red microphone.
Hello, my name is Parker, and I am not a member. I work in data at a large financial services institution, and one of the challenges that we face is convincing decision makers of the value of collecting more data. My question for you is, have you faced any challenges in convincing patients, medical institutions, funding institutions, or researchers in the value of participating with or supporting your program, and if so, how have you overcome those challenges?
Yeah, I think it's a great question. I think on every front, we're trying to show value back. And so we're trying to, now that we have enough data in hand, build data-driven arguments. So some of the studies we've done show, I probably could have done it better in the slides, that you miss a signal at a low number of participants and you find it at a high number of participants. And you can do the same thing across different populations or different ancestry groups. You can see the signal's totally missed when you don't have diversity in your sample, and when you add that diversity back in, you actually discover more and return value, particularly in variant identification. So we're trying to use data itself to drive that argument, and then we're trying to again use those balancing rocks of widely and wisely, recognizing that a really big public mistake going too quickly could really harm trust in the program, and ensuring trust is not just delivering value to the researcher, but answering every question a participant has, and ensuring to participants when they start sharing data that if you change your mind, you can stop. I think that's really resonated with people, that this isn't a one and done. We're going to continue to engage you, and if you decide to walk away, that's always in your hand, and we will then stop use of your data for research. So using data to make that argument, being transparent, and engaging with participants has been a resonating argument I think our program's benefited from.
So just to play off Larry's question in relation to the different tests, so if you're going as far back, say, as the late 90s, and then you're sort of collecting the diagnostic records for patients, moving forward, many of those diagnostic tests are not standardized. The chemistry ones are, some of the immunoassay ones are, they use, WHO standards, so that if you're using different methodologies in LabCorp or Quest or the different labs, you'd have the same reference ranges. They'd all be standardized, but not all of them are. For example, prostate-specific antigen, and there's many other examples. So if you've got the researchers going in to use the data, they're going to use AI, how are you going to ensure that they're using these results the right way, because they won't necessarily understand those nuances?
It's a great question, and we're all the time trying to learn, but also trying not to be afraid of it, because we want people to learn how to use it right. So the choices we had were to say, we only think A1Cs from LabCorp are okay, and we're going to throw the rest away. And we didn't make that choice. We said we're going to put them all in, and we're going to describe them as best we can and be as clear to researchers as we can through our data dictionaries, through our explanatory and demonstration projects, that this is a model where you, the researcher, have to think about this. This is not receiving the Framingham set where you know exactly what assay was used. This is a very heterogeneous real-world data environment, and we're trying to embrace that model and really just be very clear about it. We are not in a position, based on our lack of IRB approval, to review, for example, publications that go out. People are going to make mistakes, and we're engaging with the peer review system heavily to make sure there's as much support as possible for peer reviewers, paper reviewers, and paper writers, as well as grant reviewers and grant writers, right? So many of the ideas coming in, both at the delivery of funding, are very important to understand the study model, and then also in the peer review process, we're attempting to educate through that there's a lot of variability in the data set as well.
I'm Raj again. I'm not a member. You being a medical practitioner at some point, so given this genomic data being available, if you were to see a patient, would you be like, your recommendation or your suggestion or your conversation could be very different with a patient, because this is like giving you a 360-degree view of the patient's condition? Is that the case? If yes, how do you think this could potentially change the landscape of what data we collect from patients and how we go about treating patients, down the line, fast forward 20, 30 years. So what do you envision down the line in 20, 30 years happening? And my other question is, what is the ideal time period in terms of like a participant giving you data? I mean, is it like once a year or once every 2 years? If yes, how many years minimum you would say would be ideal for you, because somebody may leave the country or something may happen to them? So
Sure, I'll start with the second one. As many as possible. So we're asking people surveys approximately on a yearly basis. Their electronic health record data, though, we're pulling all the time passively. So active participant from the patient has been about yearly with new survey opportunities so far, but passive data acquisition, we ETL data on a quarterly basis. We're looking at doing that as much as we can across multiple health systems using claims records and other things to benefit from passive data collection. How genome works in the clinic, we're still at the beginning, 20 years after sequencing the human genome. When I see patients in clinic, most genomic data I'm using, I'm still generating for a clinical purpose. Most genomic data we use has not been population-based screening. The 20 or 30 years question is, when the cost of sequencing comes down as dramatically as it is, should you generate sequence when a child is born or before, and have that data available forever, and then have a learning system that evaluates it on an ongoing basis? I think that's a 20 and 30-year vision. There are other studies, as well as all of us, who are committed this year to enrolling pediatric participants that will start to build what that looks like. If you knew about genome from the beginning of life, what would you do differently? How would you integrate it into the clinical record? We're still at the beginning of understanding that.
So you started with a Framingham study that revealed that the understanding of the role of blood pressure was the opposite. In other words, adversity makes you stronger, but sometimes it kills you. So from this study, have there any discovery of surprises? Or it's a bit too early, and maybe should that be one of the goals, or expected, that hopefully something like that will come up?
Yes, absolutely. We're looking for those big surprises. I'd say the surprise so far has been in the depth of information in the electronic health record. While it's extremely variable across site, both how far back electronic health records go, and then how wide you can move data linkages in the passive space, is the unexplored area and the place where I think the most value will come. Electronic health records are the beginning. How much would you understand if you linked a participant's grocery card? You know, how much would you know about my family and what I ate if you knew what I bought at the grocery store? We're at the beginning of understanding opportunities of data linkages to augment data that's traditionally been available in research. I think the model's success in engaging participants is the beginning and the most surprising thing that will lead to bigger observations in the future.
Longitudinal kind of studies for an individual, but also the environment, their environment.
Yep, understanding the power of using the cloud, using the consented engaged model to broaden the data is where those new observations will come from places we haven't looked before, is a goal I hope we can achieve.
About those smart appliances. Well, I think that will be the last question, and so let us all thank Andrea for coming here and giving this talk, and for the excellent work that's being done. But before you go, I happen to have a couple of small tokens of appreciation, including a copy of Volume 1 of the Bulletin of the Philosophical Society of Washington, signed by me, but on behalf of all of the members of the society, and also a framed copy of the announcement of your lecture.
Thank you. Thank you.
And with that, yes, thank you.
Thank you. Wonderful. Thank you so much, Larry.
Thank you. Before we go, we have a few closing remarks. The 2474th meeting will be on March 17th in 2 weeks. The speaker will be Walter Harris, Chief Scientist, University of Arizona Space Institute. He will be speaking on comets, centaurs, and trans-Neptunian objects, exploring the echoes of formation in the outer solar system. The 2475th meeting will be on March 31st. The speaker will be Jonathan Losos, Director of the Living Earth Collaborative, and Danforth, Distinguished University Professor at Washington University. He will be speaking on using experiments in nature to study evolution, research on lizard island adaptation in real time. The 2476th meeting will be on April 14th. This is the usual surprise lecture, and we'll let you know at a later date who that will be. 2477th meeting will be on April 28th, and the speaker will be Amanda Padani of Cal Poly. She'll be speaking about Mesopotamian life and findings from the reading of ancient cuneiform tablets, of which there are many thousands, most of which have not been translated. The 2478th meeting will be on May 19th. It will be the annual Joseph Henry Lecture. The topic will be Building Habitat on the Moon. The speakers will be Daniel Innocente, Senior Space Architect at Blue Origin, and a former PSW speaker, and speakers from Think Orbital, Lockheed Martin, Hassel Architects, ICON, and NASA. The 2479th meeting will be on June 2nd. Speaker will be Sean Carroll, Homewood Professor of Natural Philosophy at Johns Hopkins University and fractal faculty member at the Santa Fe Institute. He will be speaking on a topic in contemporary theoretical physics. And the last meeting of the spring, the 2488th meeting, will be on June 16th, and the speakers will be Bill Merrill and Sam Brody of Texas A&M. They will be speaking about the $30 billion plus Dike project being planned to control rising tides and water surges that threaten to engulf Houston in flooding and storm damage. It is one of the largest civil engineering projects contemplated in the United States in the next couple of decades. Please check the PSW website often for updated information on meetings. Before we go, last but not least, please thank the people who have worked hard to make this event tonight and to live stream it and to record it. Thank you. Janet, Rosemary, Cameo, Robin, Robert, Lloyd, Carrie, Bill Mitchell hasn't done his work yet because he edits. So speaking of crews and those who make it all work, if you are so inclined to be helpful, and we would like to hear from you. We always need people to run cameras and help with the many things that have to be done to make these meetings occur and be successful. So if you are so inclined, please see me. And with that, I will accept a motion to adjourn the meeting. A second? All members in favor? All members opposed? Meeting is adjourned to social hour.